Hello, all you in microbiology YouTube land. Hi, Tori. How are your puppies? She got exposed. What is it that we've been going along just fine, and now all of a sudden, like a bunch of people have gone into quarantine? I don't know. Get vaccinated because I don't want to have to wear masks all the time.
The second shot kicked my butt.
The second one?
Yeah.
Yeah.
I was miserable on Friday.
I'm sorry. Well, actually, Kiara was saying that she had fever and chills and all kinds of
It
stuff.
just hurt to touch my body. I didn't even have the strength to get up.
Now, did you have a round of COVID?
Mm-hmm.
You did have the disease?
Mm-hmm.
See, that's what it is. You are, if you've had it before, your first shot is going to be like your second shot. And then your second shot is going to be like your second shot. Because
I had no effects on the first one.
that's just because you was lucky. Oh, well, when did you have COVID?
Over New Year's.
Yeah.
That was my New Year's.
Part of it is that the recommendation is something, if you've had COVID within the past 4 weeks or something, I think they're saying, no, don't get the vaccine.
Mm-hmm.
Partly because your immune system is still too primed. I mean, because theoretically, getting COVID is like getting vaccinated the old-fashioned way. And you have immunity to it. But this is giving you sort of the booster. So that's why you were having a greater response. And the first one versus the second one, you had gone long enough that your first one wasn't eliciting the full blast, but then 4 weeks later it is. Things we've been talking about. So you now have a collection of memory B cells that are going to keep you safe.
I was more miserable from my second shot than what I was when I had COVID.
Yeah, but you were, I would hope, miserable for a much shorter period of time.
But I was like, maybe I had like a runny nose from COVID for maybe 2 days.
But you did test positive?
Mm-hmm.
Well, that's part of what is the issue with this, is that some people get a very mild case, and it may be because you have a much lower viral load, or you just are tougher. And then the immune response from the vaccine is designed to be not a small load, but in fact designed to give you a full blast dose. So, thank you though for your service to your community.
Mm-hmm.
You'll be receiving a $50 Amazon gift card.
I wish.
Me too.
But if I go to Krispy Kreme Donuts, I can get a free donut.
There you go. Although generally they give you a free donut anyway. I wonder if they've actually modified that.
If you show your vaccination card, you
Right.
get free donuts.
Yeah.
Mm-hmm.
That's something.
Yeah.
Yeah, you hear that? Free Krispy Kreme donut. Of course, I guess the nearest Krispy Kreme is in Springfield.
Exactly. And I really don't need to eat donuts.
Right. Well, that's for me, is that I'm missing that. Is that it used to be my tradition, is on the final I would bring donuts. And so now all I can do is, while you're taking the final, have a donut. Or think about having a
You could
donut.
have brought them today.
What's that?
You could have brought them today.
Yeah, I thought about that, but then, no. Well, yeah, because see...
Wow.
Well, no, because this class does meet as a class, but Molecular Cell, it's only a third of the group, and so, I'm not
Don't
gonna.
bring donuts for them.
But only a third of the group. That doesn't seem fair.
I mean, you would only have to buy like 5 donuts for
I know.
this class.
But then I had to keep them secret, and then yeah, it's too much work. And sorry. Oh, which does bring me donuts. If you've got books that you got from me and you want to turn them back in, at some point, I don't know if I have a stack of tins that people gave me. No. I might
Speaking
have
of that,
one.
I got very upset with that because I went to the bookstore and bought the book for $99. And then you post saying, yeah, if you want to rent a book from me for $5, it's okay.
$10.
$10.
And did you turn it back in?
No, they don't like.
Yes, they do. I turned.
Yes,
mine
they
in.
do.
wouldn't let me.
Well, maybe it was you. You may have showed them attitude or something.
I did not.
You don't even have to ask them what you talked about. You go on the website, you click refund, they give you a whole label to print out and mail it back.
I
Yeah.
didn't do it. I went in. It's
Uh,
like, no, there's no refund.
Not a
what?
refund.
That's bullshit. Oh, should be. Sorry.
You can refund for like 30 days after.
So I guess I got a microbiology textbook that I'm gonna have for the rest of my life. So
You give me stuff. It's a good book.
So your students next semester, they want to buy it from me.
Ooh, okay. Other teaching expensive.
I haven't even opened it.
Oh. I know Jeff Pomerville, the author. Yep. When I was doing my master's and PhD, he was across the hall. And, yeah, well, he left A&M.
Person's name on those was busy.
Like Aladdin.
Well, he left A&M, went to Arizona Community College, and started working on this book somehow. And I am told that this is the #1 nursing microbiology book, and that he is like, a celebrated author for editing Alcamo's, 9th, 10th edition, and he's been doing, the new editions every few years, which is also whatever. Hey, immune system.
Yeah, we do have like some classes that are designed knowing that it's a thing that non-science majors are going to have to take those classes. They have
I wonder how many
biology
times
and
I say that.
foods that you might do experiments.
Which is my class, but there's no room for it. Sorry, there's no seats in biochemistry of food. Don't ask. Are you in it?
I am.
Oh shit!
Finally.
It's the same thing over and over again.
Mhm.
Which one are you in? Sunday, Monday afternoon, or Monday evening?
Yeah.
The Monday night 8 week.
Yep.
That's actually an interesting time.
Yeah.
Yeah.
I tried it like my sophomore year, but I was too late because obviously the
Yeah.
sophomore time to sign up for classes.
Yep. It is a delightful class, and last semester I did it, but didn't meet the lab, which made me sad, and it just was a mess. But yeah, and the Monday evening is an interesting dose. Yeah, there'll be a little bit of repeat, but with Jello. Yeah. All right. So we were going through the immune system and a little bit about various problems you can have with your immune system. Some of these being self-evident, things like immune deficiencies. That's bad news. And that one of the things that immune deficiencies do is, from a population experimental perspective, it allows researchers, epidemiologists to know and understand just how critical the immune system is for a variety of things. In part, we now know about the immune system's response to cancer cells is that T cells are able to knock back a whole range of different kinds of cancers that once you lose your T cell population after acquiring the human immunodeficiency virus that then leads to full-blown AIDS, you start to show these various kinds of tumors. And there are things that you can do to yourself. Cancer treatment is one of them, but radiation and other environmental exposures can reduce your immune system. And this is one of the reasons why people that are undergoing chemotherapy right now have to be particularly careful about, well, all the time, the flu and now COVID, because their immune system is not as tuned up as it might be. Autoimmune diseases, there are a whole range, and as more and more studies are done, the number is increasing. 46? That was probably like last year, 2 years ago. So lupus is a autoimmune disease. Some forms of rheumatoid arthritis, or rheumatoid arthritis, some of the kinds of arthritis are autoimmune diseases. There are people that begin to lose their thyroid function due to autoimmune disease, and this is known as Hashimoto's disease. Diabetes type 1 is an absolute autoimmune disease. Not really thought of it that way all the time, but it's where you have an immunological attack against the beta cells, the B cells, that produce insulin. And so you have no insulin, and very often diabetes occurs in young people, probably because you're a little bit primed for it. You have a B cell that is a little bit too close to self. It made it through that check in utero, but it still is a little close, and you might have some kind of bad flu that will kind of crank up the immune system, and that can cause these guys that are a little bit close to maybe undergo a somatic mutation that allow them to bind a little bit, And it's one of the things we talk about the autocrine interaction, is that once a B cell binds to an antigen, it begins to get a signal to cause it to divide, and so to amplify the B cell population. As the B cells are dividing, they actually have a system that allows them to have slight changes to the DNA sequence of the immune gene. And you generally don't want to have mutations being picked up by your cells. That's a bad thing. B cells, though, actually can do that a little bit. They can undergo what is known as somatic mutation. And so when that happens, if the B cell produces an antibody that can bind just a little bit better, then that one starts to divide even further and even faster. And so you have this little microevolution going on in the bloodstream as those B cells that divide faster are the ones that can bind best. The tighter they bind to the antigen, the faster they divide. And so the population of the best antibodies, at least you hope, are the ones that are starting to be built out. All right. So, as we begin to go through this then in an overview, we have the B cell which responds to the antigen. That is, you inject your vaccine. Now, in the case of COVID vaccine, just because of complexity, there are a number of different vaccine types on the market now. The simplest kind of vaccine would be, the simplest would be to take the virus and run it through the autoclave so it's heat killed, and you inject that. The next kind of virus vaccine that has been used in the past is to culture the virus over a period of time and to identify a mutant form of the virus that is attenuated, that is not able to carry out infection as well. And this is actually the basis of the two different polio viruses that nobody takes anymore anyway. And now, as modern technology comes about, we've got the ability to produce, through biotechnology, recombinant DNA technology, a specific portion of the virus, like the spike protein. So you can make the spike protein and inject that into somebody. The high end where we are now is you can modify a virus such that it will take a portion, the spike protein gene, infect cells, but it will have a receptor that is associated with different kinds of cells, not the lungs. It will be replication defective. So this is like the Johnson & Johnson virus or vaccine. It's based on a actual virus, so it is not necessarily infectious, but it will get into a cell and probably isn't replication. It is probably replication defective. It can't replicate, and it causes those cells to begin producing the spike protein. The super cool way, of course, is by just injecting the RNA into people, and it is really just a step previous to injecting the actual protein. It's just that injecting the RNA is a lot simpler to produce in real large amounts. Producing the protein just takes a lot more purification. So you encounter the antigen. The B cells then are going to be activated, the ones that can bind, and not all of them can, but one or two can, and they begin to activate. Cytokines are going to begin to actually cause them to divide more and more, and we have antibodies being generated. The antibodies are the actual thing doing something, in that they're binding to the antigen, and that helps to generate the immune response, and you can then begin to pull the antigen, the virus, out of solution. So this is what, when people are talking about antibody treatments or the production of a vaccine, is you're looking for something called a neutralizing antibody, an antibody that will bind in such a way that the virus no longer functions. The helper T cells also respond to the antigen. They are dividing, and they are providing some of the different kinds of inflammatory signals to activate the cytotoxic T cells, which are also directly attacking the antigen-bearing cells. So we've got a whole range of immune response. Now, after things have settled down, this is where you are a week or so after the vaccine, probably 2 or 3 days, week, whatever, you then begin to have the cytokines starting to slow up. The problem is that with a actual infection, as the virus is replicating, the cytokines are being expressed at higher and higher levels. And that's what's known as the cytokine storm, which is just a big inflammatory response. But you do end up with a few of the cells differentiating into the long-lived memory cells. And so you've got cells that are ready for the next response. This can be very rapid. And this is what you're dealing with when you get the second shot. So you were better primed because you had COVID the first time, but your second one, because potentially you have a really strong immune response now to it. You've been vaccinated plus. That's the positive side of it. You don't have to go through the very slow response, which is what people and what you're going through the first time you receive the infection. All right, so just to review here, the antigens, the things that you're going to make the immune response to, are being recognized as the antigen is being pulled in and then broken down. The material is being broken apart by lysosomes and various kinds of hydrolytic enzymes inside the cell, and a portion of this foreign material will get dumped into a vesicle that has the class 2 MHC, which then is going to end up presenting it. And so this is a way that the immune system knows this cell has been infected by a bacterium. With a virus, it's a similar kind of system. We have now the MHC1 that is actually presenting. The MHC is again presenting a portion of the virus. So this is the two ways we can begin to present the parts of the invading material and let cells know that, or let the immune system know that there's something going on here. So next, we have the lymphocytes, which is just the generic term for non-red blood cells. Lymphocytes are the protein, or the cells that actually are able to carry out the next part of the immune response. And so we've got helper T cells, and they interact with their receptor. And we've got the cytotoxic T cells. They're interacting with a cell that's been infected with the virus. And B cells, they are interacting, and you've got the different kinds of immunoglobulins being generated from the B cell as they begin to produce the antibody. So the helper T cells are able to do a variety of different kinds of things. And a couple of the different receptors. The B cell receptor, this is what we think of when we talk about antibodies. The T cell receptor is interacting in a secondary way. The antibody producing cells, the B cells, they recognize the antigen. And then they begin to divide very rapidly. They begin to class switch, so you have this portion of the antibody changes its actual amino acid sequence, keeping this the same. And you have antibodies that can be diffused about and begin secreted. That's how you can take a blood serum sample, spin down the red blood cells, and you have free-floating antibodies, and you can do an antibody check. What you're doing with that is you're looking to see, have you been exposed to the virus? And so, it's one of the things to think about when you're looking at virus assays. You can do an experiment in which you have on a little strip an antibody against the virus, and you're asking the question, do you currently have the virus? And so for something like COVID, you wouldn't take a blood sample, you'd spit or hakapalugi, something in the upper respiratory system. If you want to know if you've been exposed to it, you do a test in which you do take a drop of blood, spin the red blood cells out, and you're looking for the presence of antibodies against the virus in the serum. If you've got that, then that means that you are, for all intents and purposes, you're vaccinated, or at least you have a good immune response to it. All right, so now we can activate the lymphocytes. And this happens because we've got a range of cells that have bound antigens, and these cells are now interacting with other cells that have bound antigen as well, and we can begin to activate the specific cytotoxic T cells. So, one of the things that you see, like the CD8 and CD4, these are proteins on the cell surface of different kinds of T cells, and that's how we begin to categorize them. Prior to HIV, this whole area of study wasn't even hardly touched. But the fact that we now know there are different T cells with different kinds of receptor interactions, it allows us to understand what's going on a little bit better. So we then have helper T cells that are interacting. And part of what they're doing is beginning to release these inflammatory signals, things like cytokines and stuff. All right, the next step is happening in the spleen or the lymph nodes. You end up amplifying the B cells. So the B cells that can respond, that bind antigen, they begin to divide a lot. The T cells that are interacting with a macrophage that has engulfed the bacteria, they begin to divide a lot. Anyway, so you got, our immune system is keyed for really two broad classes of infectious agents. You've got bacteria and you got viruses. And they, as you hopefully recognize, way different kinds of systems. I mean, the virus only works by actually infecting a host cell and injecting its DNA. A bacterium might get inside a cell, it might just be floating around, but the virus is a whole different kind of thing, and so the immune system is sort of optimized to operate with both kinds of systems. So you're not going to generate, let's face it, infectious pus at the site of a viral infection. But a bacterial infection, you do, because you're recruiting these different kinds of lymphocytes. And we then have the killer T cells. They're able to recognize cells that have been infected with a virus, and they, in turn, can inject different kinds of proteins like perforins and the granzyme, things that can cause the cell to begin swelling up or actually direct the cell to kill itself. And this is part of what's happening when you are fighting off the virus. So, step 5, again, after the battle, we start to have the response beginning to fade back. So you're not going to maintain this high level of response forever. The T cells, the helpers, and the cytotoxics, they begin to slow up, and we have the long-lasting memory cells. And that's our immune system. So, I think for myself, talking about this in the middle of vaccinations is interesting and profound in that last semester when we were talking about this, the vaccine was known, we had it, but it only barely started rolling out. We have the vaccine really rolling out now. And so, I think a lot of us in here have been vaccinated. If you haven't, you should do it. Yes, there are stories of side effects, but for most folks, it's not a significant kind of phenomenon. That said, out of a class of 6 people, 2 or 3 of you have had fairly significant side effects. Sorry. You're doing it for public health. You're taking one for the global team. And it is a critical tool. I mean, it is the one thing we can do against viruses. If we were to have a bacterial infection outbreak, the ways we deal with that are completely different. And if you reflect upon this, we have bacterial outbreaks occur because of contaminated food, but so quickly they're taken up. I mean, the worst that happens is that Jimmy John's doesn't put sprouts on their sandwiches. Or for a period of a month or 2, fresh spinach isn't around. Now, probably most of you say, I wouldn't even care. But it is a much simpler system to begin to control when we're talking about bacteria, because the mechanism of infection is a different thing. tuberculosis, sure. I mean, it can be inhaled and people coughing and sneezing, but it is a lot less infectious than a viral infection. So we are living through a very specific time in epidemiology, and hopefully one that we won't see for a while. Again. All right, that's it. I'm out of here. Ciao. Bye. Rock on. Vinyl will drop on Sunday. When on Sunday?
That's a good question.
Yeah, that's a good question.
Morning?
Noonish.
Noonish.
Well, I don't want to have like a deadline.
Have a bunch of people emailing you.
Yeah, like, where's the David Vinyl?
Yeah.
Yeah. So yeah, how about it'll drop sometime on Sunday, day before 5. I like that. Would that help you?
Okay. Yeah.
Shit, I don't know, whatever.
Oh, thank you. Yeah.
You'll post
You'll
it
have till
4:59.
Wednesday.
No, I know.
And it's a 1-hour friggin' exam, man. All right. Well, no, the exam itself would be designed to be taken in 1 or 2 hours in class. And I never say, okay, yeah, well, that was the whole, I should probably stop this. Bye, Tori.